Hello, this is Dionisi from Université Libre de Bruxelles. In this video, I'm going to introduce a recent work entitled Proprioceptors Enriched Neuronal Cultures from Induced-pluripotent Stem Cells from Friedreich's Ataxia Patients show altered transcriptomic and proteomic profiles, abnormal neurite extension, and impaired electrophysiological properties. Friedreich's ataxia is characterized by a severe loss of primary proprioceptive neurons of the dorsal root ganglia, key components of the sensory system that provides information about body position in space and movement. In this study, we attempted to address the question of the specific vulnerability of these neurons in Friedreich through the in vitro characterization of iPSC-derived proprioceptor-enriched neuronal cultures. To these papers, we used iPS cells generated from healthy donors and Friedreich patients, along with Friedreich sibling isogenic control lines. In the latter, the removal of the GAA expansion mutation responsible for the disease was able to revert the repressive epigenetic marks at defects and locus and to restore frataxin expression. Their inclusion in the study helped us to evaluate if the removal of the GAA expansion mutation would be sufficient to fully correct the phenotype of Friedreich cells. A bulk RNA sequencing analysis was performed for control, Friedreich, and isogenic control lines at 3 different stages of differentiation: iPS cells, developing neurons, and fully mature neurons. Surprisingly, we did not detect any differentially expressed genes between Friedreich and their isogenic control lines. The gene ontology analysis for upregulated genes in Friedreich, indicating the involvement of stress-activated pathways, the response to increased oxygen levels, and the activation of catabolic activities, but also the dysregulation of cell adhesion and cellular chemotaxis. On the other side, downregulated genes in Friedreich mainly encoded proteins involved in axon development and guidance and in synaptic plasticity. We specifically detected the downregulation of 2 proprioceptive markers, the transcription factor 1X3, involved in target-specific proprioceptive pathfinding, and the calcium-binding protein parvalbumin. We also observed the dysregulation of neurotransmitter receptors expressed also at proprioceptive afferents, particularly a massive upregulation of the glutamate receptor GRIN3A, and the downregulation of genes involved in presynaptic transmission. For most identified DEGs, only a partial or no recovery was observed in the isogenic controls. Finally, we detected numerous aberrantly expressed non-coding RNAs, which could potentially contribute to Friedreich pathogenesis. Some of these elements were either massively upregulated or downregulated both in Friedreich and in isogenic control lines at all stages of differentiation. Data from transcriptomics were confirmed with the characterization of the proteomic profile of mature neurons. Top enriched pathways for differentially expressed proteins were related again to neuron projection development, cell junction organization and assembly, and synapse organization. Accordingly, these proteins were mainly localized at cell junctions, at the grocon, and at synapses. Most DEPs identified for Friedreich and isogenic control neurons were associated in a tight network and had known interactions. Although some markers of mitochondrial stress were identified, they were not those usually associated with Friedreich's ataxia. Differences detected in the transcriptome and proteome were supported by the observation of defects in neurite extension in Friedreich neurons, whose longest axons looked tortuous and twisting. Statistically significant differences were observed at the outermost level, indicating the inability of Friedreich neurites to reach the same distances of controls, probably because of their irregular trajectories. Finally, whole cell patch clamp recordings revealed some alterations in the spiking profile of tonic Friedreich and isogenic control neurons, although only Friedreich neurons exhibited a reduction of their firing frequencies in comparison to controls. A common feature was instead a delayed accommodation in response to sustained depolarizing stimuli. Taken together, our results suggest that in Friedreich's ataxia, proprioceptive neurons might not be able to properly reach and innervate their targets into the muscles or in the spinal cord, followed by the degeneration. Our data prompt a link with RANX3, a critical factor in proprioceptive pathfinding, and with the TrkC anti-free signaling, which regulates many pathways promoting neurite outgrowth and synaptic plasticity, which we found altered in our study. Moreover, the fact that isogenic controls did not show a clear recovery suggested the persistence in these lines of a homeostatic response to changes originally triggered by the GAA expansion mutation, or the contribution of other regulatory elements, as supported, for example, by similar changes in non-coding RNAs in Friedreich and isogenic control lines. Thank you for your attention. If you have any questions, please do not hesitate to contact us.